Welcome to the OTS webinar series. So today we have Eliot Morrison, and Eliot leads the bioinformatics group at Silence Therapeutics in Berlin, Germany. And this team is mainly involved in many aspects of in-silico siRNA design, including the prediction and evaluation of potential off-target effects. But he also worked on the development of machine learning-based models to address questions around oligonucleotide therapeutic development. So today, hopefully, we will get an overview of how off-target effects have traditionally been approached and explore the potential blind spots of such methods as revealed by untargeted RNAseq-based experiments. But hopefully, Eliot will also highlight the challenges associated with expanding the off-target search space, namely the explosion in false positive rate, and explores the potential solutions like machine learning to counteract it. So, Eliot, thanks for being here, and the screen is yours.
Great, thanks a lot. Thanks for the intro. So today, as Ronald said, I'd like to talk about the in silico prediction of siRNA off-targets and some of the challenges associated with doing this sort of in silico prediction. So these are our forward-looking statements. And as many of you are aware, at Silence, we develop therapeutic siRNAs. And siRNAs have a lot of advantages. For example, they have a really long duration of action. They're very safe, and they're also highly specific for knocking down target mRNA. And the specificity arises in part through the property that many off-targets are actually mediated through the siRNA sequence itself. This gives rise to some pretty interesting bioinformatics properties. And unlike, say, small molecules, where it can often be very difficult to predict off-target effects, we actually, with siRNAs, have a chance to in silico predict off-targets that are hybridization dependent. That is, they were actually governed by the siRNA sequence itself. And this is how we do this at Silence. This is a very high-level, broad strokes overview of our in silico siRNA design pipeline. And what we see is that once we've decided on a target gene that we want to develop a therapeutic siRNA against, we often have thousands, if not tens of thousands, of possible options in silico that we could choose from, essentially many thousands of potential siRNAs. And the way that we narrow this down to a more manageable number is through two parallel in silico workflows. So one on the left is the prediction of on-target activity, trying to find the most potent options. And on the right is the off-target profile prediction workflow. And here we try to find the safest and most specific siRNAs with the essentially cleanest off-target profile. And of course, what we're looking for is this sweet spot between these two outputs. So the siRNAs that are predicted to be the most potent, but also the cleanest and most specific. And it's from this intersection that we make our selections about what we actually want to synthesize and screen experimentally. And so today I'd like to focus on the right side of this workflow, the off-target profile prediction. And of course, as many of you probably know, trying to predict oligonucleotide off-targets is nothing new. I mean, people have been doing this since the very beginning. And it's broadly thought of normally in two main mechanisms. So the first is probably the most obvious. This is where you have an off-target target site that is fully complementary or near fully complementary to your siRNA guide strand. And in this way, you get downregulation of your off-target by the same mechanism as downregulation of your on-target. These in silico are relatively trivial to identify and therefore avoid, which is great, partially because you're searching with only a few edits and you're searching with a relatively large sequence. And I'll get into more details about that later. The other main mechanism that's discussed is much more tricky from an in silico perspective. This is where you have a seed match, especially a 3' UTR seed match, and therefore you can actually get a microRNA-like effect happening. And here the requirements for complementarity are much more relaxed, and this makes it a much more difficult in silico problem, with the primary reason being that the false positive rate can very easily explode. Nevertheless, these microRNA off-targets are in fact real risks that we need to consider, and they have been linked to in vitro and in vivo toxicity. So there's certainly something we need to keep an eye on. But the question I would like to pose today, kind of an open question, is this the whole story? Are these the two ways that we should look at potential off-targets in silico, or should we consider maybe expanding our search a little bit? And if we do, what are the challenges associated with that? So I mentioned that predicting in silico off-targets is nothing new, and in fact there are many very good publications out there, open source algorithms to predict off-targets. These are just two examples I'm showing. I don't want to go into the details, but in my personal experience using some of these algorithms, like I said, they're very successful when you have a high degree of complementarity. But the real struggle is in the seed matches and trying to find these possible microRNA-like off-targets, with, again, the reason being that the false positive rate can very easily explode here. This is just to say that this is very much an active field of research and development, and there's a lot of people doing a lot of good work trying to get better at this and better on our understanding of the field. So before we talk any more about in silico alignments, let's step back a minute and talk about how we can experimentally validate or identify off-targets for siRNAs. And there are two main methods that are widely used to try to do this. One is a real-time qPCR-based approach. Here, they're very sensitive methods, but the major downside, in my opinion, is that you have to define exactly what you're going to be quantifying here. So you have to, before you do the experiment, you have to design, say Tacman amplicon sets. You have to say these are the exact transcripts we're going to try to look for off-target risk from. And you have there a risk that you may miss things, especially if you rely on your in silico predictions. And the potential for false negatives means you might not look for a potential off-target that is in fact real. An alternative method would be an untargeted sequencing-based approach, something like RNAseq. And here you're not biased at all by your in silico predictions because you're in fact quantifying changes to the entire transcriptome here. And this is, of course, a good thing because you reduce or eliminate this risk of false negatives from your in silico predictions, because if you have a false negative, you'll catch it at this point with such a method. But on the other hand, this means that you're going to capture every change that's happening in the cell. So this will include not only the hybridization-dependent off-targets, but also hybridization-independent effects. Some examples might be if you're on-target biology, you knock down your on-target. Perhaps downstream, there are genes that are up or down regulated. This will be captured in such an experiment. Also, perhaps the condition of the cells. If the cells are, say, hypoxic or something, you would also capture these changes, which would be unrelated to your actual siRNA off-target profile. So the first important point here, and for people who've been in OTS for a while, of course, this is a theme, but you really need to make sure that you're designing and including the proper controls in these type of experiments, especially controls that can account for hybridization-independent effects. That's really crucial. The other important point, which unfortunately has no easy answers, but should still be considered, is the translatability of such experiments. For example, if you're interested in trying to evaluate off-target risk in a human system, you may be limited to something like a primary human hepatocyte system. And it's not guaranteed that anything you see there is going to translate very well into an in vivo study in a different species. For example, a tox study in rats, for example. Reasons being, of course, the biology is going to be different in this different species, but any potential off-target sites you see in the human system are not going to be guaranteed to be conserved in another species, of course. Likewise, you may have new off-target effects in this other species that are not relevant to a human system. So it's a very, very important thing to consider, but like I said, there really aren't any easy answers for this. So now that we have methods that we can use to experimentally evaluate off-target profiles, we can use this as kind of a base truth, and we can then evaluate how well any in silico algorithm or prediction is actually performing. In this context, we talk about predicted positives. A positive prediction here would be a gene that the algorithm has predicted will be an off-target. And a negative prediction would then be everything else. So any gene that's not predicted as an off-target, you can consider that to be a negative prediction. And then once you've done an experiment and actually measured what is happening in the cell, you can then evaluate when the algorithm got things right. So these would be the true positives and true negatives. But equally important, you have to look at where the algorithm got things wrong. And this would be the false positives and false negatives. And both of these classes have their own risks associated with them. False positives, if your false positive rate is too high, your algorithm might be a little too trigger happy and predict that every siRNA is going to have many, many off targets, when in reality, if you had actually done the experiment, you may have found they're actually very clean. So you don't want a false positive rate that's too high, because you may kick out some very good candidates in your in silico design stage. Perhaps even more important are the false negatives, and these are essentially the blind spots of the algorithm, where the algorithm says gene A is not going to be an off-target. You do the experiment, and in fact, it is an off-target. And I think it's self-evident why this is such a critical thing to keep an eye on. And it was this last category, the false negatives, that got us thinking, because occasionally what we would see when we perform an RNA-seq experiment is we would see a differentially expressed gene or an off-target that we had not predicted in our in silico prediction workflow. This, of course, we wanted to try to learn from, and we also wanted to try to improve if we could. So we wanted to take a closer look at what we may be missing with our understanding of what an off-target might look like in silico. And the way that we approached this was with a global analysis. Here we pooled RNA-seq data from more than 50 unique GalNAc conjugated siRNAs. And these experiments were all performed in primary hepatocytes at the same...
Oh, Eliot,
yes.
Sorry to interrupt. So there seem to be people that have overlapping slides. I don't see them on my screen. I assume that you also don't have any overlapping slides on your screen. So
okay,
not sure
strange.
what to do about it. Maybe just quickly restart the PowerPoint. That might resolve the problem.
I'll do that.
Yeah. Seems to be all okay from my side, but
yeah, it looked fine for me too.
Okay, let's continue from here. So people just shout out in the chat, then I will try to solve the problem if the problem keeps appearing.
Okay. Yes, so this global analysis we wanted to perform, essentially we pooled RNA-seq data from more than 50 GalNAc siRNAs from across a range of targets. And what we did is every gene that was quantified in these experiments, we did an in silico alignment to the siRNA guide strand. And as I mentioned, of course, with an RNA-seq experiment, you're not just capturing the hybridization-dependent effects, but you also have this background of hybridization-independent effects, for example, related to the target biology. And in order to get rid of these, essentially a reality check, we built a filter to make sure that we were only focusing on alignments that we believed were compatible with the underlying biology. And what we did for everything that passed this filter is we then took a look at potential patterns of different features of these alignments. For example, we looked at mismatches, how many and where they were occurring. But we also looked at things like stretches of complementarity and seed matches, and what we refer to as bulges. And so bulges are a really interesting case where you have flanking stretches of complementarity that can only base pair with the other strand if they physically push out a certain number of bases or nucleotides here. And we can consider bulges that might happen on the target mRNA side, but also bulges that might happen on the guide side. And we call these target and guide bulges. As a second reality check, we wanted to check what was possible, kind of get the distribution of these features for validated microRNA targets, because we hoped that this would give us, again, kind of a reality check about what is possible and what isn't possible in a microRNA context. And so to do this, we took 1,800 experimentally validated microRNA target sites from MIR-Tarbase, and we performed the same analysis in parallel to look for these different features. And we started with these two main mechanisms I mentioned at the beginning. So first would be these siRNA-like off-targets where you have a high degree of complementarity. And unsurprisingly, we found very, very few of these in our RNA-seq data. And I say unsurprisingly because, of course, we have already pre-screened these sequences to avoid such off-targets. And this is partially why I say it's a relatively trivial thing to do, because we, in fact, see very few of these off-targets, which is great. Perhaps more interesting is the seed matches I mentioned. And I was personally surprised to see that only about 4% of our observed differentially expressed genes have a classic 3' ETR seed match. If we relax this a little bit and say maybe the seed match is somewhere else in the transcript, we get an additional 18%. But there's still about 77, 78% of these DEGs that don't fit either of these two main paradigms, which I think is extremely interesting. I think this analysis also illustrates the false positive problem I mentioned earlier, because if we focus on the genes that were not differentially expressed, not significantly downregulated, you can see that many of them, 8% of them, have a 3' ETR seed match. And again, if we relax this, we get an additional 42% that have a seed match somewhere else in the transcript. What I take from this is that if you were to rely purely on a naive seed alignment, potentially you could have up to, depending on how strict you are, up to maybe 50% of the transcriptome being predicted as seed-mediated off-targets. And I hope it's clear from this volcano plot that we are not actually hitting 50% of the transcriptome as off-targets. So clearly there are many false positives if you rely only on this seed alignment. We then dove a little deeper into some of these perhaps more exotic features. So first, the bulges. And what we're showing here are two different types of alignments. In red are the alignments that we're very confident are compatible with our understanding of RNAi biology. And in gray are alignments that are more borderline. So some of these are certainly in silico artifacts, but others we think may be right on the edge of what we think is compatible. And we wanted to include them just to get the full picture of what may be possible. So first, we took a look at the number of target bulges that we saw in these alignments. And on the right pane is looking at the RNA-seq alignments, the siRNA alignments from the RNA-seq data. And you can see that the majority of these alignments feature no target bulges, which would be what I would expect. However, quite a few do, in fact, feature at least one target bulge, which was interesting. And this, you can see, is also compatible on the left pane with the microRNA alignments. And this, in fact, is quite well established in the terms of microRNA biology, where you can get these kind of target bulges happening quite frequently. So that was good to see. We then looked at the size of these bulges. And somewhat surprising to me was that some of these bulges are actually relatively large, I would say. Sometimes 3 or 4 nucleotides in size. And that's essentially how many nucleotides need to be pushed out, again, physically into this kind of bulge or bubble. But again, it was nice to see that this is also compatible with the microRNA alignments. And some of these are actually roughly in the same range of size as well. So what I take from this is that target bulges of this size are at least not incompatible with the whole AGO2 risk, guide strand, target site, structure, and sterics. So that's quite a nice thing to see echoed in the microRNA alignments. We also were interested, of course, in the position of these target bulges. And here we're showing the position from the 5' end of the guide strand. And here you really nicely see this requirement for seed homology here in the microRNA alignments, right? With essentially no target bulges happening within the seed here. The majority of these bulges are happening in this kind of position 8 to 10 region, which I think is quite well established, with more kind of petering out towards the 3' end. Interestingly, for the siRNA alignments, we really don't see this kind of positional dependence. And in fact, we see target bulges that happen and occur within the seed, which again was an unexpected thing for me. I'll talk a little bit more about that exact feature a bit later. So overall, it seems like target bulges, at least in some cases, in some contexts, seem to be tolerated and compatible with the underlying biology here. We then took a look at the other side. So what about bulges on the guide strand? And here the picture is completely different. We see essentially no alignments that feature guide bulges. Maybe a couple, but I would argue these are probably artifacts. We see some in the microRNA alignments, so it could be possible in that context, but for siRNAs, we really don't see any. And personally, this does make sense to me if we think of the context of AGO2 loading and the interaction with the target site. So target bulges seem to be somewhat tolerated. Guide bulges, definitely not the same level of tolerance. We then took a look at mismatches, and here we saw, in some cases, mismatches, in the range of 3 to 4, possibly more mismatches occurring in some of these alignments. But of course, it's very well established that microRNAs, have a much less stringent requirement for complementarity, and you can see this reflected in the fact that many of them have multiple mismatches. However, you also see this really nice illustration of the seed dependence here, with very, very few of these alignments having mismatches within the seed, and the majority happening more towards the 3' end. We also saw positional dependence for the siRNA alignments. Here, many with position 1 mismatches, but also mismatches towards the 3' end, which was nice to see because this has been reported before that this region is more tolerant of mismatches. Finally, we took a look at GU wobbles, which many of you are probably aware are a subset of mismatches that are more thermodynamically favorable. And our anticipation was that these would be more tolerated than any other kind of mismatch. And in fact, we were able to see that multiple GU wobbles are seen in these alignments. Interesting, though, there seems to be essentially no positional dependence. So we see GU wobbles occurring across the entire alignment, including the seed region, which again, I did not anticipate from this analysis. So this, of course, gave us a lot of food for thought. As you can imagine, it sparked a lot of really interesting conversations. But we were thinking, and I'm sure many of you in the audience are also thinking, it's interesting, but is it real? I mean, this is all in silico, right? You can do a lot in silico, but what's actually happening in the cell here? And to convince ourselves, we decided to design a validation experiment to test if one of these patterns is in fact compatible in a more generalized sense. So are these features that we should maybe look for in other potential off-target interactions? And to do this, we chose an alignment, one of these patterns that I mentioned that have this really interesting target bulge within the seed, which I think is a really interesting pattern. And we designed 20 siRNAs against a variety of targets. And we, through insertions and deletions, we altered the guide strand to force this exact interaction with the endogenous target mRNA in the cell. We then measured the impact on target activity at 2 different concentrations by a transfection experiment, and we classified the impact in 3 different categories. So the large effect group here are cases where forcing this interaction led to essentially a complete loss of activity. So these were cases where this interaction pattern was completely incompatible with actual on-target activity. But interestingly, quite a few were in this moderate effect group. And here they lost activity, but they were still active. And a smaller group had only a small effect, so only a minor loss of activity, which was very interesting and seemed to validate that, at least in some cases, Certainly not all cases, but some cases, this can be a general interaction pattern that other off-targets could in fact show. But of course, we realized that by changing the guide strand like this, we are also changing the properties of the siRNA itself. And to make sure we weren't being misled by this, we flipped the question around and said, what if we keep the guide strand intact, but we edit the target site to force this exact same interaction? And we were able to do this using a luciferase reporter system where you have complete control over what the target site looks like. And the guide strand, again, is completely unchanged. And what you see here on the left are three different siRNAs with fully complementary target sites. And you can see in all cases, they're very potent and they knock down the reporter. But on the right here are the same three siRNAs with the edited target site. So here we're forcing this target bulge interaction. And you can see that in all cases, there is a loss of activity, but they are still, in fact, active. And I should remind everyone that in the context of off-targets, even a moderate activity can still be relevant, of course, from a safety perspective, depending on the exact off-target you're talking about. So this was really nice, and it personally convinced me that, at least in some cases, this is an interaction pattern that could, in fact, show up as a real off-target interaction in an siRNA experimental context. Of course, not all the time. I meant, again, there's this group that lost activity completely, so there's probably underlying interplay with other features of these sequences, but at least in some cases. So we wanted to try to incorporate this information into our in silico prediction, but we pretty quickly ran into this issue that as you increase your search space, so for example, if you now include bulges as a possible feature to search for, then you run into this problem I mentioned already with the microRNA seed interactions, that you tend to get more and more false positives. And I hope I can illustrate this here with this in silico exercise that we performed. And here what we did is we took 10 random 19-mer sequences from the human transcriptome. Essentially, you can think of these as 10 random siRNA designs. And we aligned them to the entire human transcriptome. And we looked, what we're showing here are the number of alignments for each of these sequences. In this case, you can think of the number of alignments as kind of a naive prediction of the number of off-targets, right? And what we did is we looked at the effect of the number of alignments as you increase your search space through increasing numbers of edits. In this case, the edits being mismatches and bulges and combinations of them. And we also looked at the impact as you decrease the sequence length. And this was meant to represent the challenge of the seed searches. And the way that we can kind of understand if we're getting closer or further away from reality as we make these changes is to consider this region here I've highlighted, which is roughly the average range of DEGs that we see in an average 19-mer siRNA experiment. So any of these points where you start to get out of this dark green color, you're essentially now aligning to more transcripts than you would actually measure in an RNA-seq experiment. Essentially now you're getting more false positives than true positives in your alignment. And this is even more extreme if you consider that this is a logarithmic scale. And by the time you get to this red color, you are literally aligning to every single transcript in the transcriptome. And I hope if you recall the volcano plot I shared earlier, we are not actually hitting every single transcript in the transcriptome as off targets. So clearly, by the time you get to this range, your false positive rate has exploded so extremely that this is essentially meaningless. You have no way of differentiating between true positives and false positives. So you need a way to somehow restrict your search space or somehow differentiate these false positives and true positives. One way you might consider to do this would be to let biology guide you. So this was from a really excellent paper presented at OTS a few years ago. And here they looked at the impact on activity for combinations of mismatches at every position in 2 different microRNAs. And the regions that are highlighted in red are the regions that are very sensitive to mismatches. For example, the classic, kind of cleavage site here around positions 10 and 11. If you have mismatches here, you really severely impact activity. On the other hand, regions that are highlighted blue here are much more tolerant of mismatches, and in some cases even enhanced activity is seen with mismatches here. So what one might consider doing is use this kind of information to weight a in silico off-target alignment. For example, if you have an off-target alignment with mismatches in one of these red regions, you may consider weighting that less, because it's likely that your impact on activity is going to be very extreme. Therefore, it's less of an off-target risk. On the other hand, if there are mismatches in the blue regions, you may consider the opposite and consider weighting these higher, because these regions are much more tolerant of mismatches. The big challenge I see with this is that if you really want to consider covering your full feature space, especially considering some of these features that I was just sharing, things like target bulges, and certainly combinations of, say, bulges and mismatches, very quickly the amount of data you would need to generate to really exhaustively explore this feature space becomes really, really out of control. So it's not impossible. I mean, it's a finite amount of data, but it's a serious challenge and a serious investment to try to understand this. An alternative I'd like to propose is machine learning. So, of course, there's a lot of hype about machine learning right now, but I think in some cases, some use cases, this hype is actually pretty well warranted. For the reason being that these models can really excel, provided they have an adequate training data set. They can really excel at inferring rules that otherwise might be very difficult to explicitly define. And I think RNA-seq is a method that can generate really relevant data for training such models. On the one hand, you can generate a lot of data for a relatively small investment and resources. And the other is that you also are kind of pre-selecting for the most relevant feature space, because any features that are not actually compatible with the underlying biology should not be appearing within your DEGs, right? Because they're not actually compatible with activity. So there's a lot of potential here, I think, but there are also some challenges, of course. One is what I mentioned earlier, that with RNA-seq, you are capturing all changes that are happening within the cell. And so you need a way to differentiate between your hybridization-dependent effects and your hybridization-independent effects, because you really need to isolate those hybridization-dependent effects for training a model. And this is really not a trivial thing to do. And it needs to be considered carefully how you disentangle these sorts of data. The other big challenge is that your training data is almost certainly going to be severely imbalanced, if only for the fact that the vast majority of genes are not differentially expressed. And so your model is going to see many, many more negative examples than it does positive examples. And this can really severely impact the bias of the model and the performance. And finally, as I mentioned earlier, you really need to be careful that you're balancing your false positive rate and your false negative rate. Because if you go too far in either direction, you have certain risks associated with that, like I mentioned. Nevertheless, with all these challenges in mind, it is, I think, promising enough, and it's so promising that we have, in fact, applied it at Silence. And the way we've done that for our off-target profile prediction is that anytime we generate experimental data now, we ingest this into our model training process. And here we're able to then train new models and benchmark them to old models and see, is the performance getting better? Is it the same, or is it even worse? But we also take a look inside the black box and see what is the model actually picking up on. What does the model think is important in the data? What does the model think is not important in the data? And this allows us to actually get scientific insight into what might be happening. And it allows us to generate in silico hypotheses. Of course, this is great to set up then validation experiments and hypothesis testing experiments. I shared two of those with you earlier, this really interesting seed target pulge interaction pattern. And so we can actually pressure test some of these in silico trends that we're identifying. And of course, any of these validation experiments we perform are also generating new data. And this can then be ingested into the model training, and the cycle can continue. Ideally, hopefully, getting better every time and improving our ability to predict these off targets. So with that, I'd just like to wrap up with a few conclusions. First, as I mentioned, finding these high complementarity, high homology siRNA-like off targets for an in silico problem is actually relatively trivial. And we were able to validate that with our RNA-seq data. So this is great. We can really make sure that we're not going to be hitting these kinds of off targets. The seed-based off targets are a much more tricky challenge, trying to find these microRNA-like effects, with the main reason being that the false positive rate is often such a killer thing in these sorts of predictions. This is also, of course, really well known in the field of microRNA biology, where even finding the endogenous target sites is often quite challenging because of this exact same false positive problem. And then interestingly, in our global analysis, we found that only about 4% of the differentially expressed genes we observed have this classic 3' UTR seed match. And this increases a bit if you don't require it to be in the 3' UTR, but it still leaves about 77 or 78% of these DEGs that don't fit either of these paradigms that we were talking about. That spurred us to do this global analysis where we identified a few features that might make sense to expand our in silico search with because they seem to be compatible with the underlying biology. For example, GU wobbles and target pulges, even within the seed, seem to be tolerated. But as I hope I was able to illustrate, it's not as simple as just introducing these sorts of features into your alignment search for this reason that your false positive rate very quickly explodes. So you really need a way to either restrict your search space or differentiate false positives from true positives. And I think machine learning is a really has a lot of potential for possibly solving this problem, provided you can generate the appropriate data set. And this has the ability, I think, to possibly uncover some of these more unconventional off-target alignments that we were looking at. And finally, I just want to emphasize that everything I've talked about today is all about in silico predictions. And this will never replace rigorous experimental validation and screening process. And I think this is actually a great thing because on the one hand, you mitigate this risk of false negatives by such screening experiments, which is great, of course. But also as a bioinformatician, of course, I'm always excited when you generate new data because we can hopefully learn from these data and possibly train models and ideally improve our understanding of what these off-targets might look like. So before I end, I just want to give a quick plug in case you're not completely sick of hearing about off-targets yet. We are contributing to a subcommittee of the Oligo Safety Working Group, and we're working on a white paper right now where we're trying to provide industry recommendations for how to assess these hybridization-dependent off-target risks, not only for siRNAs, but also other oligonucleotide therapeutics. We had a poster at OTS last year that I know got a lot of good interaction, good conversations, good feedback. So if you're interested in the subject, keep your eyes open for the white paper, which we hope will be out soon. And finally, just a big thanks to the team at Silence. As you can imagine, this was very much a cross-functional endeavor. There's lots of experimental data we were able to dig into in silico. Especially want to highlight the contributions from the molecular design team, the drug discovery team, and the translational genomics team. Really couldn't have done this work without this whole team-wide effort. So thank you. And with that, I'd be happy to take any questions.
Well, thanks, Eliot, for this great presentation. I really enjoyed it. I think, we learned a lot about off-target effect. And maybe to highlight, before we maybe start. So if you have a question for Eliot, please use the Q&A function. Then I will read the questions. And maybe to highlight. So I think we have this very nice paper from Keith Gagnon and David Corey, which provide guidelines on the design of your experiments with ASOs or siRNAs. I think it's worth to read this publication and also to apply it in your own in vitro experiments. Just as a shout out. So that also leads to my first question. So because I'm very happy that you touched on taking the right controls in your experiment. But what would be the right control for an hybridization independent effect?
Yeah, let me go back to where we were talking about this. So one thing you can think about is the hybridization dependent off targets, of course, will be driven by the sequence itself. So one thing you can consider is if you are targeting a certain, target, is to test multiple sequences against that target, right? Because if you see common DEGs when you knock down your on target, this is likely, or almost certainly, not going to be driven by the sequence itself, because your sequences ideally should be completely different. So that's one strategy you can use. Make sure that you are always testing a minimum 2 different sequences against a target. Another would be to include non-targeting controls. These are really important. Something that shouldn't hit anything in your cell. And this allows you to then differentiate, say, effects from, well, possibly the chemistry of your molecule, but also the conditions of your cell, right? So with a non-targeting control, if you see a lot of things changing, you should take a close look at what's changing there and possibly consider repeating the experiment, because ideally you would want that to be clean.
Yeah, I think I fully agree on this recommendations. Another question that I have. So I think many of these RNA-seq experiments are done in control cells, or in just I think you used the hepatocytes in your experiment. So can you comment on the fact to use control cells rather than disease cells? Because when you it's quite a big step from the in-silico prediction to the patient, but the ultimate aim is to design a treatment for a patient, and then most often you treat disease cells. So why not taking cells from a patient or disease cells to do these kind of experiments?
Yeah, I think it all depends, of course, on the indication you're looking at and the availability of such cells, and also how well validated you think they represent the disease you're looking at. But certainly it could be a very interesting thing to, I would consider comparing, kind of a healthier control cell with a disease cell. Of course, for off targets, you need to consider, if you were to see major differences there, they would likely be in off-target genes that are, say, up or I guess probably up-regulated within the disease, right? So you may not actually see that many differences, depending, of course, on, again, the disease and everything. But yeah, certainly if you have access and you can validate such a system, I think you could probably learn a lot from performing an RNA-seq experiment with it.
Yep. Next question. So we go to the Q&A. Question from Jack. It's more like a practical question. So if you do an RNA-seq experiment, how long after transfection should you collect your RNA to predict the off-target effect? And if you do this, how many DEGs are expected for a highly specific siRNA?
So it's a great question. So one thing to consider is if you want to do a transfection experiment, or if all the data I was showing is from a GalNAc receptor-mediated uptake experiment. And I think transfection has the risk that the transfection process itself might lead to quite a lot of changes within the cell. So you may have a really high background of hybridization independent effects. Of course, you can control for this with the proper controls. So having a transfection control in there, especially a non-targeting molecule, can be really useful here. But in general, I think probably the more representative system, if it makes sense within the context of what you're doing, a more general system would be something like a receptor-mediated uptake. And here, and for the second part of the question, I've highlighted the rough range here of DGs that one might expect to see. So in many cases, you can find very, very clean siRNAs, but usually it's in this range that I've highlighted here. Yeah.
And to follow up, so how is the DAG defined? Is it beyond plus or minus 1 log to full change?
So this is somewhat up to who's ever doing the analysis, right? So there's no real natural threshold that you should use. I think you need to weigh the benefits of relaxing your threshold on fold change with the risk that you're introducing now more noise. So again, having the right controls in there can be really helpful because you may see kind of just natural noise in maybe 10, 20% fold change range. And so you may want to consider setting the threshold that doesn't introduce all of this extra noise. On the other hand, if you have the right controls, maybe you can remove some of that noise and you can look kind of even more relaxed on the fold change. But it really depends on what you're trying to do and essentially where you want to set that threshold.
Thanks. Question from Jan. In terms of human risk assessment, in your view, does it matter whether the transcriptomic impact of an siRNA is hybridization dependent or independent?
Good question. I mean, if it's an off-target, it's an off-target, right? So as long as you've included your proper controls and you can really be certain that it's being caused by your molecule. Like I said, sometimes it's related to your on-target biology, and then you may have to consider if that's a problem or not. I don't think there's any one-size-fits-all answer, really. It really depends on your requirements and what you need to do. It's interesting to think about what, assuming you have controls for multiple sequences against the same target, and you see hybridization independent DEGs, it's interesting to think what might be driving that beyond the sequence itself. And again, to bang on the same drum, but including the right controls can help you narrow that down.
Also fully agree again on this. So question from Randon. So nowadays there are many chemical modifications to the siRNAs. So any idea how to incorporate this into your in silico prediction model?
Interesting question. So one thing to consider would be, of course, the non-targeting control that would allow you to say, is your chemical modification perhaps inducing off targets? So with the right control, you can control for that risk. The other would be to think about what impact a chemical modification could have on these hybridization-dependent off targets. And so I think a comparison, if you have a reference, a chemical modification pattern, and something that you want to experiment with, having that reference and making that comparison should allow you to find those if they do in fact exist. Yeah.
So is it possible to add some structural data in your model? So of course, when you have a particular modification, you have a different structure of your RNA. So can you add this to the model? Would that be feasible?
In theory, yes. I mean, you can really add anything you want, provided you have enough data, to train a model on. And that would be the main bottleneck I would anticipate there is getting enough structural data for again, covering your feature space adequately enough that you feel confident that you understand the impact of changing chemical modifications. Yeah, if you have the data, I think you could incorporate it into such a prediction.
So if you compare your machine learning approach versus the alignment-based approach, how do they compare in terms of performance?
Well, I think the alignment-based approach, again, like if we really just consider like a naive alignment-based approach where you don't try to refine your search space, people of course have looked into other features, that could allow you to sort out some of these false positives and find the true positives. But essentially, the alignment-based approach, and I hope, again, I could illustrate this here, the big problem is false positives. And so the risk associated there, like I said, is that if you have too many false positives, you may end up kicking out very good sequences during your in silico design. So that's where I think the machine learning really shines, is minimizing this false positive rate.
Question from the guest. Thank you for the informative presentation. So when you knock down a particular gene, the effect is that many other genes will also be up or down regulated, just due to the on-target activity of the gene that you knock down. So how to assess this in your assessment?
Well, yeah.
On-target,
so
off-target effect.
Right. So I would challenge that is always happening. I think it highly depends on the target you're looking at and the biology of the target. So it's not always the case that you have many genes going up being up and down regulated. But again, I feel like a broken record here, but if you have controls in there to make sure that you see the same off targets or the same up and down regulated genes when you knock down your targets, regardless of what the sequence is, then I would say you can pretty confidently conclude that this is related to the on-target biology that you're looking at.
Yes. So then we move to the bulges. So I think you did some in vitro validation experiments. And how many? How big were the bulges? 2 to 5
So
were they not too big?
this one.
This one.
Yeah, so it's in this range of the 3 nucleotide bulge. So it was one of the on the large end, based on our distribution. Yeah.
Another thanks for the great presentation. Have you considered potential bulges of the siRNA itself?
Yeah, so that's exactly what we tried to show here with the guide bulges, what we called guide bulges, this one. So this would be where you actually bulge the siRNA guide strand, right? and this was where we saw essentially no cases where this was found in our alignments. So my conclusion from this is that such bulges in the siRNA guide strand are really not very compatible with retained activity.
Yes, and to follow up, again, thank you for the nice talk, Alexander. So have there been examples of siRNAs failing in the clinic, or maybe at the very final stages of phase 1 to 3 clinical trials due to miRNA-like off-target effects?
So I'm not aware, but if you are interested in the toxicity, I'd recommend checking out these studies here, because this is establishing the connection between these microRNA-like off-targets, at least in one case, and real toxicity, even in vivo tox studies.
Could you elaborate a little bit more on the machine learning models? So what are the inputs to the model? Is it just sequences or also other features? And the outputs, is it prediction of target viability?
Yeah, so the input, I mean, the training data, as I mentioned, the RNA-seq data has a wealth of information in there. Of course, we're very active in exploring different approaches to this. So we have, explored features and things. But the output, of course, is what we're trying to do, is trying to predict off targets. And so the output would be for a given input siRNA, you would have a list of predicted off targets with an associated probability or confidence in that prediction.
Question from my side. So, of course, you are building these models for silence therapeutic. So, would these off-target analysis become available for the research community? Do you keep it at silence? How to be used for academics like me?
Well, we hope to publish some of these data in the near future. We hope. In terms of the model itself, I'm not sure, that's another conversation about making that public or not. But certainly we're interested. I mean, part of this webinar was to try to start a conversation with the research community about what may be possible to look for in these in silico off targets. So we certainly want to continue this conversation.
Yes. Let's see. So a lot of similar questions. So for comparing the rank or ordering a set of siRNAs in an RNA-seq study, are you treating the cells at the same concentration?
Yes, exactly. That was here. So all of these experiments should be very comparable. So they are all treated at the same concentration and also the same incubation time.
Yeah. Question from Bruno. I think off-target effects are closely related to transcripts being expressed at a given time at a specific cell type. In your opinion, is it important to monitor off-target effect in target organs and peripheral non-target organs, or just only in the? How would you approach this?
Yeah, it's a really interesting question. I mean, you have to consider what's your targeting modality and what are the risks associated there. Of course, I think toxicologists are probably much more well qualified to answer what's relevant and not. In theory, if you have the right data, of course, you could look for possibly training models that are specific to other cell types, of course. But whether they're relevant or not, yeah, I think toxicologists could probably answer that better.
So for the liver targets, so have you seen, I assume that you compare different cell lines, so have you seen any difference in off-targets profile between the primary hepatocyte and a cell line using the same siRNA?
So the problem with other, well, say with the cell lines, is you may have to rely on a transfection experiment. And like I said, the risk of a transfection experiment is that you may introduce other effects, from the transfection process itself. So I would really hesitate to draw too many conclusions from direct comparisons. If you're comparing a transfection experiment to a receptor-mediated uptake experiment, I think you would probably be misled from these underlying impacts from the transfection itself.
So we still have a lot of open questions in the chat. So I think most of them are similar as been asked before. So what would be the biggest challenge for you in the coming, let's say, 3 to 5 years to solve?
Yeah, good question. I think, well, what I hope is that this presentation is kind of, I don't know, maybe revealed that there might be more going on, like I said, than these two sorts of interaction patterns. What I would hope is that we could generate enough data to adequately cover this feature space and ideally learn even deeper what's possible here. And then, of course, with such a data set, to be able to train a model that can even better predict off targets, obviously that would be an excellent thing to see in the coming years.
Yep, I agree on this. So there are still a lot of open questions in the chat. So I think if you have a question for Eliot, just please email the OTS. So that's info at oligotherapeutics.com, I assume. Eliot, thank you very much for your presentation and answering all the questions. I think what we learned today is there are a lot of things moving forward in this field, especially in the predictions field, but also a lot of things to learn and to take into account. So please use the proper controls in your experiment. I would say that's the take-home message for now. So thank you, Eliot, and we have the next webinar on May 16th, so same time as today. So then we have Tiberius Stam and Stefan Moravci. And they are both poster winners of the OTS 2023. So they will showcase their posters during their presentation. So thank you everyone for watching, and the presentation will be online within a few days on the OTS website. So thank you very much for joining, and see you later.
Thanks a lot. Bye.
Thank you, Eliot.